Thank you for inviting me to talk about the key characteristics. I know many of you are actually quite intimately familiar with this concept, and some it may be new to, so apologies to those who already have seen a lot of this, but I'm going to go through the concept and sort of some of the latest things that we're thinking about. I just want to put up a conflict of interest statement just in case anybody's that concerned. I do quite a lot of litigation work on behalf of plaintiffs in the United States. So a quick summary. Scientific findings are providing insights into the mechanisms of toxicity, and these are playing an increasingly important role in hazard identification. The key characteristics form a basis of a knowledge-based objective approach for evaluating mechanistic data in hazard evaluation that contrasts with and complements the reductive MOA and AOP approach. Recent IARC monograph, EPA, CAL-EPA and NTP evaluations have illustrated the applicability of this approach, and the key characteristics of reproductive toxicants, endocrine disruptors, neurotoxicants, cardiotoxicants and immunotoxicants have or are being developed. And what's really needed now is a comprehensive set of biomarkers and assays to measure these key characteristics. Why do we need key characteristics? Well, we need them for evidence integration in identifying chemical hazards. So typically we use three streams of evidence, the first being human studies, mostly epidemiological studies, animal studies, usually in rodents, of course, although we're just talking about fish, it could also be that. And then mechanistic data, which can come from either humans, animals, or in vitro studies. And we have an increasing amount of high throughput screening type of approaches in vitro studies, which are providing information, and also biomarkers in humans. And so this mechanistic data is becoming increasingly important as a stream of evidence in identifying chemical hazards. Now, the problem with mechanistic data is it poses some serious challenges. How do we search systematically for all the relevant mechanisms? How do we bring uniformity across assessments? How can one committee basically analyze mechanistic data in the same way as another committee? How can we analyze the voluminous mechanistic data efficiently? And how can, one of the most important things is how do we avoid bias towards favored mechanisms? And I'll come back to that in a little while. So the classical approach to mechanistic data has been hypothesis driven, and as scientists we all love hypotheses. It typically involves developing a mode of action, and more recently this has been replaced by the adverse outcome pathway concept, which is really a modification of the mode of action idea. The problem with that is that if you're going to regulate things, is that a mechanistic hypothesis in science is really beneficial, because if you're wrong, then that's great, because you modify your hypothesis and get closer to the truth. Whereas in risk assessment or hazard identification, this is problematic, because if you're wrong and you make a bad risk decision that cannot easily be changed, regulations take many years to actually implement, and you may have caused medical harm by doing that, and also economic harm in the other direction. And so you don't really want to have to guess the mechanism in order to make some decision. There's also a tendency of people to focus on favorite mechanisms, which may introduce bias, especially in committees. I often give the illustration that I think benzene works in a particular fashion, and another scientist may think that benzene works in a different way on the bone marrow. And depending on who's on the committee, that's going to be basically the mechanism we go with. And there are examples where the mode of action may be incomplete or wrong, and many AOPs appear to be quite incomplete. And then how many validated AOPs would you actually need for 100,000 chemical producing hundreds of adverse outcomes in different ways. So the KCs could potentially help you build unbiased MOAs or AOPs if you're needed, but you may also learn from existing AOPs about future KCs. We're currently doing the key characteristics of cardiotoxicants with a group of about 18 people, and two of the things that come up in an AOP, abnormal intracellular calcium handling and altered potassium currents, potassium ion channels. These are both likely to be key characteristics of cardiotoxicants. So the National Academy looked at the key characteristics idea and improving risk assessment in its 2017 publication, and they concluded that the approach avoids a narrow focus on specific pathways and hypotheses and provides a broad holistic consideration of the mechanistic evidence, which is really what we want. They also noted that we should develop key characteristics for other hazards such as cardiovascular and reproductive toxicity, which is what we have done. The other interesting thing from why the California EPA has sponsored much of our work is that California is somewhat unique in that it has these particular things which are actually in the legislation called hazard traits. There are 19 toxicological traits in this. There are also other traits such as potential for exposure and ability to cause environmental harm, bioaccumulation. And these are the properties of the chemicals which are the hazards. And California is theoretically supposed to make decisions based on these hazard traits. So you need to gather information on all of these hazard traits in order to really regulate chemicals in California. So these are the toxicological hazard traits in California. And you can see that at the top is carcinogenesis and genotoxicity. This is what we focused really. We have the key characteristics of carcinogens first. There's also reproductive, neurologic, and cardiovascular, and endocrine, and immunologic. All of these are being developed at the present time or have recently been published. Eventually, we hope to get hazard traits and key characteristics for all these hazard traits. So this began really with the key characteristics of human carcinogens, a very large collaboration started by IARC actually as early as 2012, that ended up being a publication in 2016 with multiple authors and produced these 10 key characteristics, which are now known to many people and have been increasingly used. So examples of relevant evidence are things like, is there something electrophilic? Does it make an adduct? Does it cause DNA damage? Does it alter epigenetics? Does it produce oxidative stress? Does it produce chronic inflammation? Does it decrease immunosurveillance and suppress your immune system? Does it bind to a particular receptor or modulate endogenous ligands like hormones? Does it alter cell senescence and alter telomeres? And does it increase proliferation or decrease apoptosis and otherwise alter cell signaling pathways? Now, many people say to me, well, these are all obvious. And as somebody once said to me on a National Academy committee, never be afraid to state the obvious. And this is the important part of this. It's meant to be a consensus, objective idea that nobody really disputes that these are mechanisms by which carcinogens can produce cancer. So the first paper was published in 2016. Kate Guyton and myself and others published then what IARC's use of it and basically a very short summary of progress to date. And we've just published a paper that has just come out in Cancer Epidemiology, Biomarkers and Prevention, showing how they're different to the hallmarks of cancer, which are the properties of cancer cells, and development of relevant assays and biomarkers to measure them. And this idea of how we measure the KCs is something we're working on right now. We've collected a group of people together to produce this paper in CBP, but we're now developing a web-based, expert-driven guide to sort of all of the assays which could measure the different key characteristics of carcinogens. We hope to do this for the other endpoints too, with the ideas of looking at what are the best tests for these key characteristics and what ones should we be skeptical of. How do you actually measure oxidative stress in people? What's the best way, and things like this. So we're trying to, that's currently one of the major projects we have ongoing. And this is of great interest actually to the pharmaceutical industry and also perhaps the chemical industry, in that they would really like an approach where they can reduce animal testing and do predictive toxicology. And this is a paper by a group from Amgen, who are very keen to develop these approaches and develop tests for the different key characteristics to modernize human cancer risk assessment and better predict the possibility of carcinogenicity in their studies. So we are not really alone in academia or in consulting or others in our desire to improve toxicity testing. And this is one approach really that I think pharma have understood could be very important and are moving forward with it. This just gives an example from of some of the data we put together. We can skip that for now, but obviously developing OECD standardized tests for things that relate to a particular key characteristic is critical. And the National Toxicology Program in the United States, led by Brian Berridge for the moment, is really keen on this idea of having the key characteristics advise them on which tests they should be focused on to develop. Some of you may recognize yourself on this picture. I need to go back, sorry. Like Tom Zoller, who was in the top right there, with Michelle Lamerle. They led the Endocrine Disruptors group on Workshop of KCs. There is Patience Brown there from OECD, Andres Kortenkamp at the back, and Gail Prince, who led helped lead the male reproductive toxicity with Xavier Arzuaga from EPA, and the female reproductive toxicants was led by Ulrike Luderow. So this group got together and they produced 3 papers producing the 3 key characteristics of male and female reproductive toxicants and the key characteristics of endocrine disrupting chemicals that Laura Vandenberg and Tom contributed greatly to, along with Michelle, in developing these papers. These are the key characteristics of endocrine disrupting chemicals as drawn by the artists at Nature Reviews Endocrinology. And not having a list of endocrine disrupting chemicals that everybody could immediately agree upon and rely upon, much of this was based on the mechanisms of hormone action and our knowledge of hormone action and how chemicals work to disrupt or alter this hormone action. And this guided the development of these EDCs. The Tom, Laura, as I mentioned, were very important in developing. So these are the key characteristics of endocrine disruptors. The top 2 are kind of the obvious ones that everybody really thinks about with hormone receptors. They think about activation of particular agonisms or antagonism to hormone receptors. There's also hormone receptor expression, of course, that could alter signal transduction in hormone responsive cells. Again, epigenetic modifications. Again, another one that would be typically people would think of would be alters hormone synthesis. But then there's the intracellular transport of hormones, blood protein expression, which alters their distribution in the body, their inactivation, breakdown and recycling and metabolism. And then actually things like hypertrophy and atrophy and things like this. So one of the conclusions that came from this, I think Jerry Heindel was one of the first to point this out, is that we have a lot of assays for some of these key characteristics, but practically none for others. And this sort of opens up, shows the data gaps that we may have in looking at endocrine disrupting chemicals. So we're currently writing a paper on neurotoxicants and developmental neurotoxicants, what the key characteristics of those. Pam Line is leading that, along with Thomas Hartung from Johns Hopkins. This has been disrupted somewhat by the COVID epidemic, but we're hoping that the paper will be submitted soon, and leading journals have expressed interest in publishing it. We're also currently working on the key characteristics of cardiotoxicants by Zoom, which is a new experience of having 20 people on a call and all talking. But it's going very well. We've broken people into subgroups, and this group has got some very experienced people on it in the cardiotoxicant field, and they are contributing greatly to this process. So how are the KCs being used and who is using them? Well, interestingly, they're being used by people like IARC. For example, their preamble now really includes the key characteristics as a way of looking at mechanistic data and making evaluations on mechanistic data. And it's now possible, on the basis of mechanistic data alone, to actually call something a possible or even a probable carcinogen. They're also being used by the National Toxicology Program report on carcinogens. There's a paper they just put out on haloacetic acids, and it's a case study. And really shows the importance of the key characteristics in their thinking. Consulting groups have started to use it, such as Toxstrategies, to look at a systematic evaluation of the mechanistic evidence for carcinogenicity of other chemicals. And then people like the Environmental Working Group, and on the other side of the spectrum, have, for example, applied them to look at PFAS in a recent publication. So they're being widely used by different groups, and certainly beginning to be more used by EPA in their evidence integration strategies. So this, we now have experience of using the key characteristics in, I think, 13 monographs, 112 to 125. This is a slide that Kate Guyton made. Just basically, actually was supposed to be presented at SOT this year, but we couldn't go. And so you can see here that quite a large number of chemicals, nearly about 50 or more, have now been evaluated with key characteristics, and we are finding out more and more about how useful they are and some of the pitfalls in using them as we apply them. So the real point about this for IARC was really that there are so many studies and so little time to evaluate them. And IARC does a working group meeting, usually it used to be in person, over 9 days, and they would look at all the epidemiological studies. Typically in that, you only have tens to hundreds of studies, usually actually only 4 or 5. You have studies of cancer in animals, it says tens of studies. Often I've been on IARC committees where there's only 1 or 2 or 3 long-term cancer bioassays, but there are thousands of papers to look at on the mechanistic data, 3,000 for PCBs, 2,000 for benzene. So how do you actually read 3,000 papers, and what do you do? So, and how do you even search for it? So we've been working on search terms and systematic measurements of all these different search terms. We want to bring uniformity across the assessment, so we want to have procedures that people can follow to think about whether this evidence is strong, weak, or moderate. How do we analyze this voluminous database efficiently, and how do we avoid this bias I spoke of earlier? So we've developed a set of keywords and mesh terms. These are being actually revised currently by Luping Zhang and Kate Guyton. Are being expanded and used more. And you then can put those into your favorite organizing program. This can be, it's Hawk, it's called Hawk. It's an open software at IARC, but there are other systems like SysRev, which uses AI to organize these things. But the way that this works is you're able to look at the relevance of the literature. This is for 1900 benzene. And then you break them down into key characteristics, and you'll find that there are a relatively smaller number of papers then that you can assign a subgroup of 2 or 3 people to look at and make decisions about and bring the whole thing together quite efficiently. So it facilitates this review. It may replace or assist in the development of MOAs and AOPs and networks. And as I mentioned, it may actually improve predictive toxicology and molecular epidemiology for disease prevention if we can develop the appropriate assays and biomarkers to measure them. And this is the idea again that we need, basically the big thing that needs to happen now is the development of tests, or the description of already existing tests and their assignment to particular cases, and understanding that we're looking at the complete picture when we test chemicals. I think, for example, for endocrine disrupting chemicals, we may actually be missing a lot of possible effects on things that are not usually tested for. And this may be true also for environmental chemicals for cardiotoxicity and perhaps others also. So to finish, basically, one thing that we have noticed is that there, and probably you also, is that there are overlaps in between male and female reproductive toxicants, EDCs, neurotox, and key characteristics of carcinogens. We have noticed that, for example, epigenetics alterations commonly comes up, as does oxidative stress and inflammation. And receptor-mediated effects are obviously altered as a cell proliferation. And so this suggests that there may actually be a common set of key characteristics of bioactive hazardous chemicals. And so if you think about how this could work, you could think of about the idea of there being 2 tiers of testing. One being, if we take tests for these particular endpoints that we're talking about here, all these key characteristics that are common to many types of toxicity, we could start with that as a first tier and say if it gets past that, we could then do a second tier of tests that are specifically for things like developmental neurotoxicity or endocrine disruption. And so we could have multiple tiers of tests that actually informed us very quickly about the hazards of chemicals. And this is a paper that we're working on right now that shows the associations between the key characteristics of carcinogens in C, endocrine disruptors in E, female reproductive toxicants in F, and male reproductive toxicants in M. Some of them have no correlations with other types of KCs, but you can see that there is a great deal of overlap in the top left corner of KCs and in these different types. And if you look at something specific like epigenetics, you can see the correlation with epigenetics in endocrine disruption with that in carcinogens, in male and female reproductive toxicants. So there's direct interaction. Similarly for receptors, which we know is important in cancer, the 8th key characteristic, this links with 4 of the endocrine disruptors, the first female reproductive toxicant, KC, and the male one. And similarly in hormone signaling, looking at that from female number 1, you can see that there are multiple connections to the endocrine disrupting chemicals. And so there are shared and common key characteristics, and there are also unique ones for these different things. And this has implications, I think, for toxicity testing moving forward and understanding the hazards of chemicals. And with that, I want to thank you for listening. I'm happy to talk about this and discuss with you, answer any questions. I want to thank the California EPA, especially Lauren Zeiss, for their support of this, which was also supported by the NIHS Superfund Research Grant that I have, especially Catherine Guyton of IARC. And finally, I want to thank some of you who are on this call, and also all the people who have contributed so far, completely free, completely without any compensation, except for economy class travel in coach on Southwest or something like that. There's been 90 people, 43 institutions and 11 countries to date that have participated in this. I think this shows the great deal of interest in this particular approach, and also the willingness of people to give their time in developing this approach further, for which I'm very grateful. And thank you again for inviting me.